right? As far as the nervous system and stuff, okay? It's a derivative of the amino acid tyrosine. Now, anybody know what are some other ones made from tyrosine? The catecholamines, epinephrine, norepinephrine, and also thyroid-producing hormone, TRH, making that. So it's very small and has 3 amino acids in it. And it is a neurotransmitter, but it's also considered to be a hormone, okay? Its principal function in the hypothalamus is that it inhibits the release of prolactin. What's prolactin do? Milk let down, all that good stuff, right? And so basically, its main influence over the hypothalamus is over the secretion of prolactin by the pituitary, as inhibition, as we say. And if the hypothalamic control over the pituitary is lost from a tumor or something like that, then basically you're going to lose almost all your pituitary hormones except prolactin. Okay.
So I thought that oxytocin did the letdown and dopamine just created the milk itself. I mean, and prolactin just created the milk itself.
Prolactin does help to create the milk, but if you don't have the milk, what does it do to have milk letdown? Okay. And so it will help. It causes the mammary epithelial cells to secrete this type of thing. And that will be either caught up in the secretory cells or in the ducts. And then when you have oxytocin, that constricts the members of the muscle layers and forces it down. Okay. They usually work synergistically together. All right, let's look at this case. Now, this picture is not of a doll with no rear end. Okay. That's just for some reason the picture looks like that. What's that dog doing? All right, this is a 5-year-old male castrated mixed breed dog. Was admitted with a 2-week history of listlessness and decreased appetite. When examined at the clinic, the animal was dehydrated, lethargic, showing neurological signs. And this is standing in the corner, staring at the wall or head pressing. And at anisocoria. What's anisocoria? Okay, and this is just showing you what example of anisocoria there.
Wait, what's it?
Anisocoria, different size.
Okay, different sizes.
Of the pupils there. All right, y'all didn't have that as good anyway.
Okay.
All right, pain was elicited on flexion and extension of the neck, and an intracranial mass was suspected, and the owners opted to euthanize the dog. Okay, now, one thing I have a problem with this is that I would like the intracranial mass be verified before owners would opt to euthanize the dog, but you got to do what the owners want to do, right? Now, is it legal to say, yeah, I'll euthanize the dog, and when the owners leave, you give it to somebody else? No, you can get in trouble like that, okay? But anyway, we did the necropsy, and the gross diagnosis, the carcass is in good body condition. Significant changes are restricted to the brain skull base. In other words, the bottom of the brain up there. The left image below is a soft pink irregular mass measuring approximately 1 by 2 by 2 centimeters present in the ventral surface of the brain, and it is basically centered over the diencephalon slightly to the left of the midline. So they're talking about basically this area over here.
Yeah.
Okay, the mass extends caudally from the olfactory tract to the rostral margin of the pons, which basically takes up the whole hypothalamus on that side, doesn't it? And laterally, the mass abuts the piriform lobe on the right and covers half of the piriform lobe on the left. Now, it's been fixed, and the other slide on the right there shows you a cross section, and y'all see the mass in there, right? Everybody see it? It's kind of reddish color, so it makes it a little easier. And does that look like it's putting pressure on the brain up above it and everything? Yes, it is. Okay. Well, the image below is after they took the brain out. And what should be down there on the bottom of the skull? Shouldn't the pituitary be down there? Pituitary should be right in this area right here. You see it? No. So it appears irregular. The floor of the skull appears irregular, thickened, and slightly yellow. And you couldn't really identify the pituitary gland. And that doesn't mean there's none of it there, but it's not that nice, discrete, sex tissue that's supposed to be. Okay. So you think this animal, does that explain some of the neurological signs this animal's showing and everything? Yeah. All right. Well, basically you did histopath on it, and this is what this one looked like. Do y'all remember what the normal's supposed to look like on the hypothalamus? So that's why I put that in there so you can see what it's supposed to look like. But you can see, this doesn't look like this tissue right here, does it? And you can see, what's it doing to the tissue above it and around it? It is compromising it and impinging upon it, isn't it? Well, this is what a normal hypothalamus should look like, with the axons, the neuron cell bodies, and the fibers and stuff in there. And this is what this one looked like. Does that look normal? No, you can see there are 3 cell types comprised of mass. The main cell type consists of nest and solid, lobules of loosely packed neoplastic cells separated by collagenous septa. And these cells are round to polygonal and with distinct cell margins. A scant to moderate amount of flocculent amorphocytic cytoplasm in the central round nucleus with granulated coarsely clumped, often chromatin, and an indistinct nucleolus. And what are some neoplastic signs? Do you get increased nucleocytoplasmic ratio? You also will get some prominent nucleoli present. So these aren't quite as prominent, but again, look how much they're proliferating. You see any of those nice neuronal cell bodies and things in there like we saw in the normal one? So that's a problem, isn't it? Now, another cell type had a little more vesicles as far as their cells and the cytoplasm are concerned. And they're large polygonal cells, moderate amounts of eosinophilic cytoplasm, large oval vesicular nucleus with a single distinct nucleolus. And they often have discrete cytoplasmic vacuoles. And if you look down closely, you can see these cells. Some of them, pointer doesn't look like this one right here. You can see the vacuoles in the cell and everything. So it's another one of the cell types. And as far as the last one, you have some trabecula. And you can see how you've got these cells lining up right here. So you get these palisaded cells in there. So there were the 3 distinct different cell types. All right, so based upon all that, the moderate amount of anisocytosis and anisokaryosis, and again, anisokaryosis is one of the most prevalent things you'll see as far as neoplastic change is concerned. All right, we got all those cells in there and everything. So what's the most likely morphological diagnosis of what's going on here? Think it's a neoplasm? Okay, you say what kind? All right, this is malignant mixed germ cell tumor, so it's a canine subcellular germ cell tumor. We say that because there's different cell types present. Generally with a neoplasm, what does a neoplasm mean? It's malignant or tumor, either malignancy or growth of the same cell type, usually, isn't it? And then we can identify it. But what if we're back involving some cells produce a lot of different type cells, don't we? So what if we're in that germ cell, what if we're affecting that? Are we going to get different cell types affected? And that's what we got here. So what special stain can you put on this a section of this tumor to make sure that's what it is?
Acid fast.
Okay, it is acid fast. What else would we use acid fast for?
Alphabetic.
How about tuberculosis, mycobacterium? Also, you'll see it with nocardia and actinoplasmosis. But anyway, got that. All right, well, we do that. Is that stain positive? Yeah, when we say acid fast for mycobacterium, it's a different type of acid fast stain. So the stain stains pink. This is out of fusion. This stains this gold brownish color. And so, putting all that together, what's your interpretation then? This is an acid fast stain section, neoplastic shell, so multifocal, moderate to strong cytoplasmic positivity for alpha-fetoglobulin. This confirms the diagnosis. Prognosis, well, you know what prognosis for this dog is, isn't it? So what if the animal wasn't dead? How long would it be alive? Not long. So what would we call this, LOL? Yeah. Well, we know he has lack of longevity since it's a necropsy, a dead dog disease, right? But anyway, but even so, in other words, the option to euthanize was probably the most humane thing to do as far as this particular one is concerned. Okay? Now, as far as, I'm not going to read this to you, I'm going to read this. This explains about this tumor and why it's causing these problems in these areas. You all can read this as fast as I can read, so you all go ahead and just make sure you're familiar with that because it does help explain what's going on. Okay? All right. So that's the hypothalamus. Okay, well, let's look at the pituitary. Okay, here's just some examples of the pituitary. Another name for the pituitary is like hypophysis cerebri, or hypophyses cerebri, however you want to pronounce it. And you can see that it's not really a very pretty organ, but it's very important. And it's very similar as far as growth and histomorphology. It's very similar among species. This pituitary, you can see the location. See the hypothalamus above there. And it hangs below the hypothalamus. It's on a stalk, which is the infundibulum. And that's basically the part of the hypothalamus that bends down that whole forms the stem that the pituitary dangles on. Okay? It sits in a pocket of the basal sphenoid bone. What's that called? Sella turcica. And what's the membrane that holds it in there called? Diaphragma sella. You can read the slide, it tells you there on it. And in other words, so that membrane, when it's intact, remember when we took that brain out of that last dog, we couldn't find the distinguished pituitary? But during that membrane will make it, the infundibular stalk break, so the pituitary stays in the cella turcica. But it didn't in that because it had been damaged, so it wasn't normal retained like it would be. Okay. Now, it's divided into two major subdivisions, and they each have different embryological origins. The neurohypophysis, also it's called the posterior pituitary. Its components are the pars nervosa, which is the biggest part down there, the pituitary stalk, which is again the yellow coming down, and the median eminence, which is part of the hypothalamus. And what neurohypophysis is, it's an extension of the hypothalamus. Okay, so it's a neuroendocrine portion. The endohypophysis, or the anterior pituitary, it's histologically it looks just like a normal endocrine organ. Okay, it's got the epithelial cells and all this type of thing in there. Its main component is the pars distalis, which is the biggest part, the pars intermediate, which is between it and the pars nervosa, and the pars tuberalis, which doesn't go all the way up as far as the the pituitary stalk does, but it goes up part way. And what it is a way for the vessels to come down that are going to basically anastomose onto that part of the pituitary. Okay. This is just showing you these sections in the bovine pituitary gland. I know it doesn't look like that picture because instead of looking at it head on, we're looking at it sideways. That's why it doesn't look round and stuff right there. And that's just showing you the different parts. Now, it's very difficult to look at a gross specimen of the pituitary and say, well, this is this and this. But, this is just showing you as far as the slave one. Here's a human pituitary. Which one's the nervosa? This one over here on the, y'all looking at us on the left. Okay, the distalis, the anterior pituitary is on the right on this one. Okay, here's a feline. And notice how much, if you looked at the human, the biggest part was the anterior pituitary, wasn't it? But when you get to these other, the posterior pituitary, when you get to these other organs, it's a little bit more that size discrepancy. But again, you can see the light colored area, that is the pars nervosa in there. The thing that goes around it is the pars intermedius, which is part of the anterior pituitary, and then you see the pars, distalis down at the bottom there, this area over here.
What about that yellow, like to me it's yellow, shaded area? Like the slant?
Yellow shaded area.
Do you see this
Down
part?
here?
Like that, yes.
Well, those are vessels and things coming down.
Okay.
So the capillary plexus and some stuff.
Capillary plexus.
That's why they're a little yellow, because the red cells are giving that the stain. All right, so remember the relationship between the hypothalamus.
Is there a specific reason why you see a more distinct pars intermedium in cats rather than humans?
Yes, it is. You also see it more distinct in cats than in dogs, because a lot of the intermedius is much more functional in the cat and the horse. And one of the problems is that very often, particularly in the horse, when you have tumors of the anterior pituitary, the chromophotoadenomas that secrete ACTH, they occur in the intermedius because there's so much more activity there. And that's a lot closer to the hypothalamus, isn't it? So it has more of a propensity to cause more neurological problems in the horse and the cat also. So just, now, don't ask me why it was decided and, that they should do that, that's, have to go with evolution. I don't know when it got decided like that, okay? All right. So this is just showing you what we remember the hypothalamus. As far as the neurohypophysis is concerned, remember the neuron cell bodies are up in the hypothalamus and the paraventricular and supraoptic nuclei. They send their axons down through the pituitary stalk down into the neurohypophysis where they have their axonal endings in there. And there are blood vessels, major blood vessels all throughout that area. Okay? Whereas if you look at the anterior pituitary, those neurons are also up in the hypothalamus, but they will have their axons will be around the capillary plexus that comes in and will feed down into the anterior pituitary, and so it delivers those stimulating or inhibitory hormones into the blood. Blood vessels carry it down to the anterior pituitary, where it releases them around the secretory cells there, and then will release whatever they're supposed to release. So there's a difference. Remember that the anterior and posterior pituitary are different as far as how they produce and give off their hormones. Okay? There's actually no endocrine cells in the posterior pituitary, neuronal type thing. Again, this is from the textbook, and it just shows basically what we just talked about. This is the schematic illustration of the relationship between the hormone synthesizing areas of the hypothalamus and the pituitary gland. Because remember, what controls allows secretion by the pituitary? The hypothalamus. And that's why I say, it's actually the queen behind the throne because it runs everything. Okay? Okay, now as far as how they develop embryologically, as we said, each of the major divisions of the pituitary come from two separate primordial parts. If you look at the neurohypophysis, it forms from a diencephalonic evagination, an outpatching of neuroectoderm that extends downward from the floor of the third ventricle. And it expands as the axons of the cells in the diencephalon grow down into it. So that's why it is a neuroendocrine organ. Okay? And forms the neurohypophysis, as we say, which consists of those different parts we talked about. But again, when you look at the section of that's why it looks so pale in the histo section, because it's axonal cells and axonal endings with microbial cells around them. So you don't see these secretory epithelial cells or anything down in there. Okay, as far as the adenohypophysis is concerned, it forms from Rathke's pouch. You all remember Rathke's pouch? Okay, so it's an outgrowth from the area of the roof of the embryonic oral ectoderm, and grows toward the infundibular process on the floor of the diencephalon, and they join, and they're joined forever as life and get together, okay? And it forms the glandular parenchyma of the adenohypophysis, and again, it's the pars distalis and pars intermedius, the main components that it forms. Now, a portion of the pouch will extend around the infundibular stalk, and that forms the pars tuberalis. Not a whole lot of importance given to the tuberalis, but it's just a portion in there. This is just showing you a diagram of how the development occurs. And you notice the adenohypophysis loses its connection as it develops from the, Rathke's pouch, while the neurohypophysis maintains its connection. And you can see why it would. If the neurohypophysis didn't maintain its connection, it couldn't do anything. If you break the axons, nothing's going to be coming down or anything. So that's why we got that. Okay, now as far as the histology, again, the neurohypophysis, since it is an outgrowth of the CNS, it basically resembles the neural tissue that you see in the brain. So it's about like microscopy. So you see fibers, neuroglial cells, and then the axonal endings. Now the axonal endings do expand, which we'll see in a minute, and they form what are called herring bodies, which are just accumulation of the neurosecretory products in the end of the axon, the synaptic area of the axon. As far as the adenohypophysis, you can see on H&E, perhaps you get what we call 3 cell types are distinguished, and we call acidophils, basophils, and chromophobes. In other words, we're using an H&E stain. Those that stain blue are basophils, those that stain pink are acidophils, and those that may or may not stain at all are your chromophobes. That doesn't mean chromophobes won't have any stain, but they won't be quite as distinct as you'll see with those other cell types. If you use, now you can't look at those and say, oh yeah, this is going to secrete this hormone, this is going to secrete that hormone. You can't do that by just looking at a routine section. For that, you have to do immunohistochemistry. It's where you can test and find out exactly what those cells are secreting by those techniques. Okay? Now, as far as the cells, they also call them by their cell name. So after you, what they secrete, so after you do a histochemical stain, you can figure out what kind they are. Now, what do you think the somatotropes secrete? Growth hormone, somatotropin, growth hormone. What about the lactotropes? Prolactin. What about the thyrotropes? Thyroid stimulating hormone. What about the corticotropes? ACTH. Okay. And what about the gonadotropes? FSH and LH. Okay. And again, when you do histochemical staining, you can figure out which one to do what.
Excuse me.
And they're not any really specific area. It only has one cell type. They're interspersed throughout the gland. Okay. Now, as far as the vascular supply, the arteries of the pituitary come from the internal carotids. Carotids is the inferior and superior hypophyseal arteries. The superior one goes directly up to the hypothalamus. As you can see up there, where the superior is coming in up at the top. And that supplies the blood supply. Basically, that's the main thing that supplies blood to the hypothalamic tissue. Okay. Now, if you look at the, pardon me, the inferior, it's the one that will supply the cells down there in the neurohypophysis and also goes over into the adenohypophysis. Now, this is not the supply that they're going into the hormones into. This is for their life. Okay, it supplies the nutrients and things for them. And then you can see that the superior hypophyseal arteries will also branch